So let's discuss question number 127. The vascular cambium normally give rise to. Vascular cambium results in the formation of the secondary xylem and secondary phloem. So if we look into the first option, phalloderm, so you have to keep this in mind. Phalloderm is formed by the cork cambium. Second, primary phloem. Primary phloem ये प्रोकेम्बियम बनाता है. Secondary xylem, yes, this is the work of the vascular cambium. V is vascular here. And periderm. So you know the cork cambium? Cork cambium on the outer side. On the outer side, it leads to the formation of corky layer, and on the inner side, it results in the formation of the secondary cortex. These three layers together constitutes the periderm. So vascular cambium results in the formation of secondary xylem. So this is the right option. Question number 128. This is related with the DNA replication. We have to look for the option which is correct for the Okazaki fragments. So during the DNA replication, you know what happens, the two strands of the DNA, they separate. Right? So this is the template strand which has the polarity 3' to 5'. Right, here the primer would be added. Yes, this is the RNA primer, and now after the addition of the RNA primer, now the DNA synthesis will occur continuously in the 5' to 3' direction. So this strand which is formed continuously, it is the leading strand. But if we talk about the second strand which has 5' to 3' polarity, here the DNA fragments, they are formed, it is formed discontinuously, and the DNA fragments which are formed, they are known as Okazaki segments. Now here the primer would be added on this side. Yes, this side the primer would be added. Right, and after the addition or after the formation of the primer, now the DNA synthesis will proceed in 5' to 3' direction away from the replication fork. So if we talk about the leading strand, the synthesis is towards the replication fork, and this is the lagging strand where the Okazaki fragments would be formed. Here the DNA synthesis will occur away from the replication fork. Right, so now we can look into the options. Yes, the leading strand is formed towards the replication fork. So Okazaki fragments, they are associated with the lagging strand, so it is no way related. Even this statement is also associated with the leading strand, so it is also not related. So only there are two statements which are associated with the lagging strand. So here the lagging strand towards the replication fork, no. Now coming to the fourth option, the lagging strand away from the replication form. Yes, this is the right option. So the right option for this question is option number 4. Now let's discuss question 129 here.